Greetings everyone, and welcome to another Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Reaching Beyond the Data to Prevent Target Identification Failure. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. It has become relatively common knowledge that bringing a new drug to market is a slow and expensive endeavor, costing between $1.5 and $3 billion and taking up to 12 years. Yet, what many people might not be aware of is the fact that 90% of drugs entering into clinical trials fail before ever reaching the final launch phases. While this failure can occur due to a multitude of reasons, the deficiency of the drug to show efficacy towards the intended target is the leading cause of failure in Phase 2 and Phase 3 trials, with poor compound selection also being a major contributor to the lack of drug success. Let's meet our speakers for this GEM webinar, who will describe some new tools and techniques for target identification and lead detection that harness the power of diverse data sources, advanced analytics, and visualizations. Our presenters are confident that with these tools, investigators should be able to eliminate failure due to inaccurate target identification, allowing for improved preclinical validation as well as better lead compounds. Our first speaker will be Richard Harrison, Chief Scientific Officer at ClaraBate Analytics. Dr. Harrison will provide a brief introduction into current target identification modalities and point out areas where improvements could aid researchers during the drug discovery process. Our second speaker will be Carlos Ferriman, a senior scientist and consultant. Dr. Ferriman will describe a specific case study for one of these new tools, target druggability, and how it can be used to identify targets for the neuromuscular disease myasthenia gravis. Our final speaker today will be Matthew Wampole, a senior solution scientist at ClaraBate Analytics. Dr. Wampole will also provide data on target druggability case study about potential shared targets for the autoimmune disorders Rheumatoid Arthritis and Chigorin Syndrome. Before we get rolling into Richard's presentation, I want to encourage everyone to submit questions for our Q&A session at the end of all the talks. We will try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All right, with all the particulars out of the way, let's get our webinar started. Richard, the audience is listening.
Hello, I am Richard Harrison, Chief Scientific Officer at ClaraBate Analytics, and I would like to thank you for joining us today to discuss reaching beyond the data to prevent target identification failures. The pharmaceutical industry has faced enormous pressures over the last few decades. Declining revenues, a constantly changing regulatory landscape, and increasing payer pressures have forced all companies to identify processes to increase innovation and productivity. This has led to a mixed view of the state of the industry. These pressures are highlighted in the industry trends in 2016. Starting at the top left, productivity, as measured by NCE approvals, was lower in 2016 for any year since 2010. This has also led to an increased use of priority designations, signaling a changing regulatory environment. The bottom left highlights payer pressures and the increased voice of the customer in the continuing increase in healthcare costs, particularly in the U.S. It's not all bad news. In the bottom right, we are witnessing the improvements in the science behind understanding diseases as illustrated by the realizations of the promise of harnessing the immune system in fighting diseases such as cancer. At the top right are the business results from all of the above-mentioned pressures. In 2016, the value of deal-making in the industry decreased, while the total number of deals was the highest on record. All of this points to an industry in constant motion. And at the heart of it all is the fact that drug discovery starts with the identification of new targets. I will try to convince you in the next few slides that the most important aspects of drug discovery is identifying targets for diseases, and that picking the right targets will have a profound positive effect on all of the pressures the industry is currently facing. Let's begin by looking at the current state of clinical trials. According to the Centers for Medical Research, in the year 2015, The probability of success moving from phase 1 to market is less than 10% across all therapy areas. In fact, compounds entering phase 1 between 2008 and 2010 and being assessed in 2013 showed only a 7% success rate going from phase 1 to market. Efficacy is a reason for failure in more than half of phase 2 and phase 3 trials. And in the middle is data published by ClaraBate Analytics scientists that show on the left-hand pie chart, the purple area indicates greater than 50% of all phase 2 and phase 3 trials fail for efficacy. And the pie chart on the right-hand side shows the breakdown by therapy area. And as you can see, it's across all therapy areas, with oncology being the highest. In a more granular study, scientists at AstraZeneca showed that 40% of their projects fail in clinical trials because no clear link is made between the target and the disease. And an additional 29% fail because the compounds chosen did not have the correct physical properties or did not reach the target tissue. So in summary, we're failing 9 out of every 10 times. We know that for at least half of the failures, efficacy is the reason for failure. And we know the things we need to do in order to increase that chance of being successful. Researchers currently use a variety of methods to identify and qualify new targets. The most popular of these is the mining of scientific literature. Other methods include biochemical studies comparing normal versus disease, genomic methodologies, such as differentially expressed genes for normal versus disease states, genetically predisposed populations, and finally, real-world observations are all currently used to identify new targets. And at the heart of all of these methodologies is the use of previous knowledge. As a drug hunter for over 30 years, I am aware of the difficulties in identifying the best targets for further research. At ClaraBate, we have spoken with many of you about these difficulties. And from your feedback, we have developed a series of interacting applications to better enable drug hunters by gathering information from a variety of sources and placing that information in easy-to-use tools. At the top right is a screenshot of the Key Pathway Advisor, which uses omics information to identify potential targets based on differentially expressed genes. These identified targets can then be analyzed in the applications we will focus on today, Target Drug Ability, or TD, an app that enables researchers to find all relevant information about a target, including structural information, genetic information, known compounds that interact with these targets, competitive intelligence, and animal model results, all to understand the complete target landscape. These tools allow researchers to rank potential targets and choose those with the greatest chances of success. And combined with additional ClaraBate products, our speakers today will demonstrate how using Target Drug Ability, they were able to find and rank targets, accelerating the identification of life-saving medicines.
Excellent. Thanks very much, Richard. That was a very nice overview and a great introduction for our next two speakers. Before we begin Carlos's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. We'll try to get to as many questions as we can, but send in all your questions as soon as possible. All right, let's continue with the presentations. Carlos, the floor is yours.
It is a pleasure to share with you my experience with Target Drug Ability. I am a drug hunter with experience in competitive intelligence, and I will show you today how TD can help you with the target selection. Today, I will focus on a disease called myasthenia gravis, and entering this disease on the TD landing page is very easy. And as you can see, TD provides multiple synonyms for this disease. Before we continue, I would like to explain briefly what myasthenia gravis is. An autoimmune disease where antibodies either block or destroy, for example, the nicotinic receptors at the nerve-muscle junction. This attack by antibodies leads to skeletal muscle weakness. Of course, there are other proteins attacked by these antibodies, for example, the muscle-specific kinase. Let's turn our attention to the results from TD for myasthenia gravis. 132 targets are represented here as bubble plots. Firstly, we use binary classifiers that indicate the presence or absence of specific data, and these results are shown here in red. To be more specific, targets that have experimental pharmacology data. The second one is targets that have launch or active drugs. Furthermore, targets with biomarkers. But it is also possible to cluster the results by target families. For example, which of these 132 targets are enzymes, channels, receptors, and they are here shown in blue. An additional key feature of TD is the ability to rank and filter targets. This is really a very important aspect of TD that can help tremendously save time running a competitive intelligence analysis. We can control, to a certain degree, the amount of risk, say, moving from complete novelty with high risk, little is known about those targets, through condition novelty, and down to early development, where there are some molecules or biologicals known for this target. This means for the early development that there is less risk, more is known, but the counterpart is that the field might become a bit more crowded. And each avenue offers multiple targets with varying degrees of risk and/or opportunity. For example, here I am showing the results of choosing from the initial page of 132 targets. I'm choosing animal models, yes or no, so narrowing it down, together with the option of complete novelty. And this narrows down the search to a single target, in this case, IL-2 receptor subunit beta, and you can follow the pink band. Alternatively, if I choose targets with experimental pharmacology and narrowed down by a condition novelty, which is in this case I chose cancer, this narrows down the search from 132 targets down to 2. And they are indeed alpha-6, beta-2, nicotinic receptors, and skeletal muscle troponin follow, in fact, in this diagram, the 2 green bands. This is a key feature of TD. You have to realize that we started with 132 targets, and after filtering and ranking the resulting targets, we end up only with 3 targets. This is truly remarkable. Let's consider a different scenario where I would choose targets already in early development. So once I choose that, TD ranks these targets, and for example, I could delve into rank number 7 in this case, is called saxin. Just briefly, saxin is a protein highly expressed in neurons and it is responsible for some forms of spastic ataxia. So let me turn your attention to key information I can get for Saxin. On the left-hand side, there is a sunburst diagram that quickly allows me to find out what information is available for this target in connection with myasthenia gravis. And in fact, I find a few entries in another ClaraBate Analytics database called Integrity, where I can delve into these molecules. Furthermore, I'm alerted to a genetic evidence reference that is a genomic analysis of patients with myasthenia gravis. All in all, the ability to jump, so to speak, from the TD tool onto multiple databases of information is a tremendous advantage of TD. So now let me turn your attention. There are multiple links available within TD, and in fact, in the case of Saxin, I was able to access the 3D structure of Saxin and also information about existing known binders. So through these links, TD allows me to explore, on the one hand, crystal structures of Saxin, and I found one structure of a domain of Saxin in complex with a small molecule. I'm also alerted to the presence of known Saxin binders, which is a potential starting point to bootstrap a process in an early research project. So I will turn your attention now to a focus on targets with marketed drugs. Before I move on, the bubble diagrams can be clustered in terms of the different families. In this case, acetylcholinesterase is a very well-known target that in connection with myasthenia gravis, and we can quickly analyze the known inhibitors of acetylcholinesterase for this very disease. As you've seen, acetylcholinesterase has many drugs associated with it, and TD offers the opportunity to analyze the drugs, for example, by organization, which company is it coming from, by the type of experimental pharmacology available, for example, is it a KI, an IC50, or is it an MIC, and also by the mechanism of action. This information is readily available within TD, and once again, it can be very useful in terms of understanding the target in connection with the disease. Acetylcholinesterase is a well-studied target with multiple drugs, but what we want is to focus on prior art molecules or biologicals in specific for myasthenia gravis. And TD provides the sunburst approach, which enables us to focus very quickly on drugs specific to myasthenia gravis. TD allows us to highlight drugs marketed for myasthenia gravis. And in fact, in this figure here, we see pyridostigmine bromide, and it's very easy to go from TD onto Integrity, which is part of the ClaraBate Analytics arsenal of databases. And we can quickly delve into this drug. The 2D structure is readily available, as well as a wealth of information, including patents, literature papers, organic synthesis, and clinical trials. TD provides multiple links to key databases, for example, EMBL, OMIM, and the Protein Data Bank. In this case, I illustrate with an example of a crystal structure of acetylcholinesterase in complex with a potent reversible inhibitor C-547. And this example illustrates the time saved to find relevant information for this target and for this specific disease. So in this case, I am sharing with you the crystal structure of C-547, a known inhibitor of acetylcholinesterase for the treatment of myasthenia gravis. This is one of the most potent and selected reversible inhibitors of ACHE with a long residence time, approximately 20 minutes, and this is very suitable for the treatment of MG. This makes C-547 a promising drug for the treatment of myasthenia gravis. And all this information is readily available from TD. Let me summarize. Target druggability provides users with new links between targets and diseases, multiple strategies with low or high risk, and it also enables users to navigate easily through complex data to improve the decision-making process, and equally importantly, to save an enormous amount of time. I wish to thank my colleagues, Montse del Fresno and Leo Lafferty-White, for the opportunity to use TD and to share my views with you. And I thank you for your attention.
That was a great presentation. Thanks, Carlos. I think our audience now has a greater appreciation of how these new methods for target druggability can really simplify the drug target identification process. Before we start our final presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Again, simply type your question into the Q&A box on the left-hand side of your screen and hit submit. We'll try to get to as many questions as we can. All right, Matt, time to bring the presentations home.
Thank you, Jeff. My name is Matthew Wampel, and I'm a solution scientist here at ClaraBate Analytics. As a solution scientist, I help the customers answer their scientific questions using our products and content. For today's presentation, I'll be talking about shared therapeutic targets between rheumatoid arthritis and Chigorin Syndrome. Now to start, let's look at a little background about rheumatoid arthritis. This disease is an autoimmune disease where the immune system attacks the joints. It causes inflammation and painful swelling. Without treatment, it can lead to bone erosion and deformity. Studies have indicated that in developed countries, the prevalence of rheumatoid arthritis is between 0.5% to 1% of the adult population. Of those affected, women are 3 times more likely to develop rheumatoid arthritis than men. And even with treatment, roughly 80% of these patients become partially disabled after 12 years, and 16% are completely disabled. This makes finding new targets to treat rheumatoid arthritis a major need in preventing the pain and suffering of these patients. To complicate suffering with autoimmune diseases like rheumatoid arthritis, have a chance of a secondary disease called Chigorin Syndrome. Chigorin Syndrome is an inflammatory disease that affects the exocrine glands like tears and saliva glands. This leads to also painful swelling and the disruption of normal function of these glands, causing things like dry eyes, dry mouth, and other such incidences. Women, again, are 3 times more likely to develop a more aggressive form of this disease than men are. And as many as 4 million Americans are impacted by this disease, and current treatments are really focused around the symptoms of Chigorin Syndrome, but don't necessarily treat the underlying disease. So being able to find a new target for therapeutics that can both treat rheumatoid arthritis and are also beneficial for the treatment of Chigorin syndrome would be a great boon to these patients who are suffering from such debilitating diseases. To start with, we would need to gather information about the potential target we would want to develop a drug for. The primary question you'd be starting with would be, how connected is the target with the disease I want to treat? But what about other questions that I need to think about when developing a drug for this target? I need to know if someone else is already developing a drug that would compete against me. Are there animal models already developed related to this target that I could use to test my therapy on? Have any other binding studies been done to show that this target is actually druggable? If I have a list of targets I'm interested in, can I prioritize them to help decide which to start with? To start the search, you might go out to the literature and look for the latest research of this disease. While this can help educate you on the latest information on the field, it can take a lot of time to go through and read each of these articles in literature. So we could go to other open source repositories collecting information about targets and disease. But really the question is, are you getting all the information that you can need to answer the various questions, to stay informed and make decisive decisions? At ClaraBate Analytics, we've been manually curating information from a variety of sources such as journals, press releases, clinical trials, conferences, and patents for many years. From this wealth of information, TD connects the potential targets with therapies in development for disease, different literature evidence connecting the target to the disease, as well as animal models related to a disease. The platform provides interactive visualizations to allow you to see and filter your targets, the ability to rank those targets based off of their novelty or competitive information, and to dive deeper into the biological significance of these targets and the roles they play in signaling pathways. The goal is to really help researchers and those target hunters find the relevant targets for the disease, rank them, and provide them all the information they need to assess the druggability of these targets. We begin by entering rheumatoid arthritis and Chigorin syndrome to have target druggability find all targets related to them. It looks for information on drugs, disease evidence, and animal models linked to both of these diseases, then reports back on all of the associated targets for both of the conditions I'm interested in. The results of this search start off as a landscape of targets that match our initial criteria. I have 147 targets that were returned that have a relationship between rheumatoid arthritis and Chigorin syndrome. The data that's being displayed initially is related to all the content associated with these targets, so all drugs, all animal models, all genetic evidence. Each circle here represents a specific target. The coloring indicates the highest phase of development of these drugs related to each of these targets. The target is grouped by the different families they are related to, and the size of this circle indicates how many drugs are associated with the target. Now, I might have a more specific question, so I can use different filters and change the visualization to really fit my needs. Since I know that Chigorin syndrome doesn't have as many therapies out there in development as rheumatoid arthritis, I can use a filter to trim down the displayed results to just those that are related to Chigorin syndrome. I can also change this visualization to more focus on some of the disease relationships between Chigorin syndrome and these targets. Here, the targets are now grouped depending on if there is a biomarker associated with them. So on the left are those targets without biomarkers related to Chigorin syndrome, and on the right are targets that do have biomarkers related to Chigorin syndrome. I also changed the size of these circles to be representative of the count of genetic evidence related to each of these targets, and again, with the particular disease that I filtered for, Chigorin syndrome. So from this visual, there are some potentially interesting targets for Chigorin syndrome, but how can we start prioritizing these targets without individually clicking on each one and looking more detail about it? is a table now. This table allows you to see all the values and all the record counts that we have associated with each of these targets, depending upon if it's related to the drug, an animal model, genetic evidence, or the biomarkers. Additionally, this table allows you to begin to rank these targets based off of the different pieces of information related to the target. So the first thing I might want to do is really look for some novel. The different rankings allow us to choose options on how we want to prioritize the targets based on the risk we want to take in the drug target. Here we are looking at a list of targets that have been prioritized for complete novelty. That means that there are currently no drugs in development for these targets that we know of. This will allow you to look at these targets and develop without worrying about competition. Now, if you want a little more confidence that this target is actually druggable and has a potential ability to impact diseases, we can use other filters such as condition novelty, which will find targets with drugs that are not currently in development for a particular condition of interest. Or alternatively, we can look at early development, which prioritizes targets based off of drugs that are in early development up to phase 1. So we can have some confidence of the target's ability to modulate disease, but we might not have to worry about as much competition yet. So let's now again focus on Chigorin Syndrome and those novel targets that we have pulled up. By filtering for Chigorin Syndrome, I find that there's 2 targets that are potentially interesting. Transportin 3, which I find 7 different genetic evidence records associating it with Chigorin Syndrome, as well as COE1, which has 2 biomarkers related to it. So let's take a look a little bit more about what I can learn about Transportin 3. We can visit the target's homepage to learn more about the records associated with it and learn more about its biological significance. Here we see that Transportin 3 is a nuclear transport receptor that is important for the import of various spliced factors to the nucleus. We can also find the sunburst diagram that shows all the curated records that are associated between this target and different diseases. Focusing on the sunburst, we can find the specific genetic evidence records that link this protein with Chigorin Syndrome and rheumatoid arthritis. Not only do we see how many records are associated with these diseases, but we can also find the specific articles that we curated relating the disease with this target. You see the 7 records that I previously mentioned for Chigorin Syndrome, but for rheumatoid arthritis, we have additional 6 genetic evidence records linking this particular target with rheumatoid arthritis. So it's pretty interesting for a novel target. Seems to be a lot of research around it, but no one's really developed a drug for it yet. So maybe it's a little questionable how druggable it is, especially if it's a receptor around the nucleus. So let's look at targets that have a little more development related to them. So this time, I ranked my targets based off of early development to see what targets have some drugs in early development up to phase 1 related to them, and what information we have related to Chigorin Syndrome on that target. Interestingly, one of the drugs, the interleukin-7 receptor subunit alpha, or IL-7RA, has 6 biomarkers associated with it, and it actually has 2 drugs in development for Chigorin Syndrome. So that's pretty interesting, and this could be an interesting target to go after or become a part of its development of. So again, going to the home page for IL-7RA, you can see that there's a lot more research devoted to this target. So there's more information available on the target homepage, associating the target now with other content areas. There are different drugs that have been tested on it, biological pathways in our pathway maps that can show the significance and what role this particular target plays in signaling cascades, as well as the different genetic evidence associating this target with different diseases. So let's first take a look at those two drugs related to Chigorin syndrome and learn a little bit more about them. We can take a little bit more about those drugs, see what phase of development they're in, how they're associated with that target, and if there's been any pharmacological data reported about them. I was interested in one of these drugs, as one is being developed by GSK, but the one MD707 is a drug I'm not too familiar with. So there's a little blue box that you can click. It'll take you to another platform we have called Integrity, which is a scientific database on drugs in development, collecting various pieces of information about the drug and published scientific data on its development. Here I can learn a little bit more about who's developing this drug, what other diseases this drug is being related to. But I can also see what some of the latest information that has been published about this drug is. Interestingly enough, over the past year, there's been a rapid succession of mergers, collaborations, and even a worldwide licensing agreement with this company, and specifically for this drug we're looking at. So this could be a very hot topic of people who are interested in targeting IL-7 for treating these diseases. Now let's take a look at processes related to IL-7RA. We can click on the pathway maps, and we get back a list of those different processes that are related to IL-7RA. Looking through the list, I see a lot of relationships with different immune processes, immune response processes, and how they're related to IL-7RA. We can click on one of these maps and learn a little bit more about the relationship. See how IL-7RA really starts this signaling cascade at the top, circled in purple, and then follows through a series of interactions that can activate JAK, STAT, which is a well-known pathway for immune response. So potentially by targeting and inhibiting IL-7RA, this would be a good way to maybe inhibit some of this overactivity in the immune system related to rheumatoid arthritis, as well as for Chigorin syndrome. But I have the question of, just how related is this pathway in general with rheumatoid arthritis and Chigorin syndrome? If I do hit IL-7RA, are the targets downstream of it important in rheumatoid arthritis, or would it really have no impact to other information have related to those diseases? Answering that question is a simple. We can take all those targets from the pathway, put them back on the landscape view, and begin to filter the relationships of those targets with the diseases we're interested in. For rheumatoid arthritis, quite a few of these targets are related to it and already have a number of drugs targeting them. On the other hand, Chigorin syndrome actually has 3 targets, one of them, of course, being from IL-7RA, but we do see one of the novel targets that we previously looked at, CLE1, is among the targets that would be impacted by inhibiting IL-7RA. So overall, inhibiting this particular target could be very beneficial to both rheumatoid arthritis and Chigorin syndrome patients, and potentially have some synergies with other targets that would affect it downstream of IL-7RA. Target drug ability provides a number of advantages for researchers looking for novel targets. It can save time by combining records on biological, experimental, and competitive information into one platform, provides analytics to rank the novelty of a target based on its disease evidence and competitive intelligence. It also provides various ways to visualize the content as interactive graphics to enable researchers and target hunters to focus on the content that is most relevant to them in their search for new targets. And now I'll turn it over to Richard to wrap up today's webinar.
I would like to thank our speakers for these interesting case studies. I would like to thank you all for joining us today. We hope you saw the power of target druggability to enable your drug discovery research. As a drug hunter, I am truly excited about the Target Druggability app, and I can honestly say that this app would have saved me weeks to months of work searching for the information I needed to pick and rank targets. I look forward to hearing your success stories using Target Druggability in your research efforts. If you would like more information about Target Druggability or anything else you heard about today, please reach out to us at ClaraBate Analytics. And at this time, I would like to turn it over to our audience for any questions on what you had heard today.
Thanks for those final notes, Richard. And Matt, thanks for the great insight into the connections between the two autoimmune disorders and how ClaraBate's advanced analytics can streamline the drug discovery workflow. So before we start the Q&A session, I want to remind everyone that this is your final chance to submit questions for our speakers. So hurry up and send them in now. We already have a lot of questions that have come rolling in, and some of them look really great, but we're giving you a few more minutes to send in your questions for all of our speakers. Give us a few moments on our side to get everything situated, and we'll begin the Q&A presently. All right, everybody, so let's start with the Q&A. We have some really great questions that have come in. So first question is going to be for Richard. Richard, one of our audience members would like to know, with all the tools currently available, why is the rate still less than 7%?
Oh, that's a very good question, and I think one that every pharmaceutical company, both large and small, struggles with on a very regular basis. I think the simple answer is drug discovery is a very difficult process. And even though we've been doing it for quite a long time, we continue to learn as we go along. And the hope is that we're taking those learnings and put it in every time we do new projects. I'd also say that knowing what needs to be done and implementing it aren't always the same thing. Sometimes we know what to do, but we just have a hard time getting that into processes. And then finally, I believe we are starting to see some small increases. In data ClaraBate Analytics published in last year in Nature Reviews, we're able to show that phase 2 studies, going from phase 2 to phase 3, the success rates increased from 20% in the years 2010 to 2012 to 25% in the years 2013 through 2015. Although it's only a 5% increase, we see the same thing in phase 3, going from about 55% to 58%. So while they're not drastic changes, I think we're starting to see an uptick in the success rates, and we hope that's going to translate into higher success rates across the entire drug discovery paradigm.
All right. Thank you very much, Richard. Carlos, we have a question for you. One of our audience members would like to know, what differentiates target druggability from other tools you've used in the past for target identification?
That is a great question. Thank you. There's a couple of things that are very important here. There are many new links between targets and diseases. There are multiple strategies with low and high risk. It is very easy to navigate through complex data, and you can access multiple databases from TD. And finally, a key point is the tremendous amount of time saved.
All right. Thank you very much, Carlos. Matt, we have a question for you. One of our audience members asks, what potential updates are there for target druggability in the future?
That's a really good question. So, we're always taking a lot of feedback from our customers on what kind of information they need in their pursuit of new targets and novel targets. But we already have a lot of ideas that we're starting to implement, including better ways to filter our data, how to improve the ranking algorithm that we're already using in target druggability, as well as new ways to visualize the content that we're providing to make it easier for our various researchers and target hunters to find those targets quickly and efficiently without having to waste time navigating around through different tools and tabs to find that information.
All righty. Thank you very much, Matt. Richard, back to you. We have a question here that says, could you provide more details about where the information in target druggability is actually coming from?
Sure, I'll take a start at that question, and I'll ask Matt to join in afterwards as well. The information from target druggability comes from a variety of publicly available data sources that were curated by ClaraBate Analytics curators. The sources include literature, which is a large basis for many of the things, as well as information from clinical trials, publicly available information from brokerage reports, press releases, a number of different sources. To be more specific to the ClaraBate world, it comes mostly from Integrity, with information supplemented as well from Medicor and a number of our Cortellis products. And I'll ask Matt to further elaborate on that.
Yeah, certainly. So we have a team of annotators, PhDs, MDs who have experience in the field who are constantly monitoring these various sources of information. Like we should say, clinical trials, poster presentations at different conferences and congresses, FDA regulations, and just keeping an eye on what drugs are in development, but also trying to better understand the biology that's going on behind a lot of these targets and how they're important for different signaling pathways. And so it's from these various databases that we are collecting this information, all manually curated, providing it through our platform here known as Target Druggability, just to make it easier to find those targets, both the well-studied and the more novel targets that are just being discovered.
All right. Thank you very much, Matt and Richard. That was a great answer. Carlos, another question for you. Audience member would like to know, is target druggability done in real time? Or conversely, how long does it take?
Yeah, that's a great question. I would say that within half an hour, you can get a sense of how to run the tool. It's very intuitive. And from there on, you can spend maybe another hour, but it's done truly in real time. It is very intuitive, and you can get quickly to the set of answers that interest you the most.
All right. Thank you, Carlos. Matt, question for you. Do you have a software or other automatic tools to do ranking, filtering process?
Certainly, and this is something that's always of interest to researchers, a way to automate their processes or tools to help them in their research. So that's one of the big features of target druggability, is we already have an algorithm built into it to allow you to take a list of targets you might be interested in and rank them using various pieces of content that we've made available through this platform. As far as other tools, I mean, we are at ClaraBate Analytics a fairly broad company looking at the various areas of research in drug development. So other algorithms we have available come from tools like Medicor, which is a more systems biology pathway analysis tool. But we also have different consortiums where we work with different organizations to develop and refine various algorithms that are already out there called computational biology for drug discovery. And those are the two prominent ways we have different algorithms to help filter, rank, or analyze data for target pursuits.
Great. Thanks very much, Matt. Richard, back to you. One of our audience asks, I'm currently an Integrity customer. What would I gain by purchasing target druggability as an add-on?
Well, one, you would gain the analytics associated with it. Another thing you would gain is information that does not currently exist within Integrity. So as I mentioned earlier, we pull information not only from our Integrity tool, but from Medicor as well for some of the pathway analysis. And we pull from some of our Cortellis tools for some of the information around clinical trials or information associated with drugs that have actually gone into the clinic. So you will gain more information. You will gain all of that information in one very easy-to-use tool. And I think those are two key things that make it more interesting than just Integrity on its own.
All right. Thank you, Richard. Matt, question for you. I think this is a good one here to kind of bring it back around again. One of our audience asked, can you please summarize what is target druggability and how to access it for trial?
Certainly. So target druggability is a content source for all the information you need to identify or find new targets that you might be interested in for treating a disease. Be the simplest way to summarize it. You know, we had talked previously about different content sources that we're curating. providing you that information, and then visuals and ranking algorithms to determine what the most interesting and potentially useful targets there are for potentially treating these various diseases that may have an unmet need. It can be from seeing the competitive intelligence around a target, understanding the biology around a target a little bit better, or just seeing if there's something brand new that's being discovered for a particular disease and its biological relationships. As far as getting access to the trial, we will have a survey at the end of this webinar. So if you're interested, for those attending today's webinar, just let us know through that survey if you're interested, and someone will reach out to you and get in contact about how you can get access to target druggability.
All righty. Thank you very much, Matt. Richard, this will be the last question for you. Do you have similar tools to help me with the next step of the process, i.e. dealing with drugs under research rather than targets?
I like that question very much. And the simple answer is yes, we are developing a number of other tools. So what we've done when we started to develop these applications is we started with a very early target identification, and we had tools such as our Key Pathway Advisor, which I mentioned earlier, which uses omics analysis. We then take those different targets and we put them into target druggability to help sort and rank the targets that are of interest. The next two applications we're working on, and these will be released sometime, one later this year and one very early next year. The first one is something called drug design. Now, drug design is more for medicinal chemists who can go in there and see all of the compounds that are currently out there against a particular target and start to pull together some sort of SAR around those targets to get a better feel for how to deal with those targets from a chemistry standpoint. So a lot of the cheminformatic information is in there. And then the last app we're putting together is something called the DMPK Viewer, where we can pull together all of the DMPK information from all the different species, animals and humans, into one place, all normalized into the same sort of units. So it makes it infinitely easier for somebody to take a look at all the information from a DMPK standpoint across all species in one place. So the idea is to be able to get you started with a target, help you identify lead molecules for that target with our drug design API, and then finally help you pick the best animal models using the DMPK Viewer.
All righty. Thank you, Richard. And it looks like we have our final question for Matt. Very straightforward question. Matt, how often is the database updated?
That's a good question. A lot of our customers ask us all the time. So our annotators are always reading new articles, going to see new presentations, checking out different patents that are being published. So we're constantly curating this information and then adding it into a database once these pieces of information have gone through our quality control checks. So generally you see information being updated in target druggability pretty often. So daily or so from the different content sources that we are curating.
All right. Thank you very much, Matt. And with that, we've come to the end of our webinar. So I'd like to remind everyone that the webinar will be archived on our site for up to a year. That's at www.genengnews.com. So if you missed parts of it, you can watch it that part again, or feel free to forward it to your friends and colleagues, which we always like to recommend. I'd like to thank Richard, Carlos, and Matt again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to ClaraBate Analytics for sponsoring this webinar. So hopefully we'll see you again in the near future at another Gen webinar. Goodbye for now.